The overall goal of this procedure is to identify the most optimal 2D crystallization conditions for the structure determination of membrane proteins by electron crystallography. This is accomplished by first using a compatible detergent during purification to solubilize the protein from the native lipid bilayer. The second step of the procedure is to add exogenous lipid and remove detergent via dialysis of the protein-lipid detergent mixture, resulting in 2D crystals under carefully controlled conditions. The third step of the procedure is to flash freeze the samples into a vitreous state. The final step of the procedure is to collect data by cryo-EM. Ultimately, results can be obtained that yield the final structure of the protein through computational image processing of the data.
Hi, I'm Gengxiang Zhao from the laboratory of Ingeborg Schmidt-Krey in School of Biology at the Georgia Institute of Technology.
Hi, I'm Dreaden from the Berry Lab in the School of Chemistry and Biochemistry and the Schmidt-Krey Lab in the School of Biology at the Georgia Institute of Technology.
And I'm Matt Johnson from the laboratory of Ingeborg Schmidt-Krey in the School of Biology at Georgia Institute of Technology.
Today we will show you a procedure for assessing 2D crystallization trials by EM.
We use this protocol in our laboratory to identify and optimize 2D crystallization conditions.
So let's get started.
To begin this procedure, negatively stained samples on carbon-coated 400 mesh copper transmission electron microscopy, or TEM, grids are prepared. Pipette 2 microliters of the proteoliposome sample onto a carbon-covered TEM grid and incubate for 60 seconds. If the